Hello everyone, welcome you all in the another video tutorial. So in this video lecture, we will discuss about the hemagglutination or the phenomenon of hemagglutination. So now let's start with the introductive part. So basically, hemagglutination is a specific form of agglutination that involves the red blood cells in their phenomenon. So basically it has two common uses in the laboratory, which are blood typing and quantification of virus, which is also called as hemagglutination assay or hemagglutination test. So before moving to the hemagglutination, let's understand first that what is the agglutination. So basically the word agglutination means clumping of particles. In the biological processes, the agglutination is the process which occurs when any antigen is mixed with its corresponding antibody and the antigen or antibody particles are clumped with each other, and this process or this phenomenon is called as isoagglutination, and this process is known as agglutination. So basically when the agglutination is complete or it involves the red blood cells in its process, this phenomenon is known as hemagglutination. So as we have discussed in our earlier slide that the process of hemagglutination is widely used in the laboratory in two different uses. The first use of this process in the process of blood typing, and the another use is viral hemagglutination assay, which is also used for the quantification of viruses. So now let's discuss about the first use of the process of hemagglutination, which is named as blood typing or blood compatibility testing. So basically the blood compatibility testing also includes blood typing. We know that what is the blood type. So basically blood type or blood group is a classification of blood. We know that all the organisms consist different types of blood groups. In the case of human, there are different blood groups like A, B, AB and O. So these blood groups are differentiated on the basis of the presence and absence of several antibodies, and these antibodies are present on the surface of red blood cells of those organisms, and they are diagnosed by the presence of several antigens, and these antigens may be some proteins, glycoproteins and glycolipids, or sometimes some carbohydrates. So basically the antibodies bind to A or B blood group antigens present in the sample, and the blood typing involves determining the ABO and RH types, which are blood group types. So the testing of these blood types is used to diagnose and to prevent several complications like pregnancy complications, which can occur due to the different blood group of baby and mother, which causes several complications or growth of the baby. So it also used are also tested before the blood transfusion in any patient to prevent several complications due to the different blood groups of the patient and blood. Now the blood compatibility test make the use of reactions between blood group antigens and antibodies. So we know that the ability of several antibodies to cause RBCs to clump together, and they can bind to antigens on the cell surface, and this is called as agglutination. And when the condition involves RBCs, this condition is also called as hemagglutination. So here the process of hemagglutination is used for the diagnosing of different blood groups in human and in other organisms also. So basically there are two types like ABO and RSD types are diagnosed in the blood typing. So now let's discuss about the another use, which is called as viral hemagglutination assay. So basically it is also a type of assay or testing used to the process of hemagglutination, and this was developed in 1941 or 1942 by an American virologist named as George Hist, and his method is used for quantifying the relative concentration of viruses, several bacteria, or several antibodies in any organism. So, the principle for the viral hemagglutination assay is based on the ability of certain viruses which have the ability to bind with the RBCs or red blood cells through their proteins which are named as glycoproteins present on the surface of those viruses and bacteria. So this assay uses this ability of those microbes to quantitate these viruses, and this testing method named as hemagglutination assay is used for the quantifying or quantitative testing of those viruses. So basically, when the RBCs are coated with antigen to detect the antibodies in the serum, the test is called as indirect hemagglutination test. Now let's discuss about the method of the hemagglutination assay. So basically this method used a 96-volt microtiter for the diagnosis or for the procedure of this method, but there are several rapid tests available for the hemagglutination assay, and there are some materials used for this method or for this procedure, including a glass microscopic slide, or it can be also a ceramic slide, a 10% suspension of RBC, micropipette and posture pipette, PBS, which is called as phosphate buffered saline, which is used as a buffer solution in different types of biological researches. Now, the another component or material used is negative and positive control allantoic fluid samples, and the sample which is used for the diagnosis. So these are the materials used for the hemagglutination assay. So now let's discuss about the steps or the procedure for the hemagglutination test. So these are the steps of rapid hemagglutination test, and this test have the ability to determine the presence of hemaggglutinating agent in the sample within the time of 1 or 2 minutes, and that's why it is named as rapid hemagglutination test. So the steps include placing 4 drops of RBCs in the glass slide or a white tile, or after that to each drop of the blood or the RBC, 1 drop of the control and test samples are used as follows. So the first step includes 1 drop of PBS, which is named as phosphate buffer saline, and after that second drop is used for the negative control allantoic fluid which have no hemagglutinine, and the third step include dropping a positive control allantoic fluid which contains hemagglutinine. Now the fourth drop is of an unknown sample which is to be tested. So these drops are made or dropped over 4 drops of RBCs, and after that they are mixed by rotating the slide or tile for 1 or 2 minutes, and then they are observed and the results are recorded. So basically, when we compare the results or the observations made by these tests, we can see, so these are the results which can be observed after the method or after the steps, and the results include the agglutinated RBCs or red blood cells which have the clumped appearance, and that's why they are easily differentiated from non-agglutinated RBCs. The RBCs which are mixed with the positive control allantoic fluid will clump within the 1 minute after the method completes, and those RBCs which are mixed with the negative control allantoic fluid and PBS, which is phosphate buffer saline, cannot clump and they remains as in even suspension. So after the judging the results of the test sample and comparing those results, we can distinguish between the positive and negative controls, and that's why here the phosphate buffer saline and the negative allantoic fluid controls are used to detect the clumping of RBCs and absence of virus, and the positive control allantoic fluid is used for the presence of the virus. So these are the results made by the hemagglutination assay.